Okay, it's 10:15 and we can start. Welcome, warm welcome everyone to the public defense of Pongprecha Malaluang, which I will call PAC from now on. My name is Bodil Strömholz and I am deputy head at the Department of Clinical Science, and I have the great honor to chair this event. Antimicrobial resistance, or AMR, threatens the effective prevention and treatment of an increasing range of infections caused by bacteria and other microbes. AMR occurs when bacteria or other microbes change over time and no longer respond to medicines. Antimicrobial resistance is a global challenge. Antimicrobial resistance in horses in conjunction with assisted reproduction. That is the title, and that is the topic of today's thesis or dissertation. And this thesis is accepted for public defense at SLU in accordance with the legal requirements to achieve doctor's degree in veterinary medicine. And to fulfill these legal requirements, we have a number of key persons here today. First of all, we have the respondent, PAC, who is here, and we are really looking forward to your presentation in a short while. Then we have another very important person, and that is the opponent, Professor Graça Ferreira Dias from the University of Lisbon, Portugal. Then we have the ones that decide actually after this, and that is the examination committee. And then we have on link, Professor Amanda Marjorie de Mestre from Cornell University in the United States. Very welcome, nice to see you there. We also have Docent Bart Gardella from Utrecht University in the Netherlands. Welcome to you too. And we have Docent Maria Kareskoski from Helsinki University. Warm welcome. We have the reserve Bengt Gus from SLU, but as you all are here, he is not here. He decided not to come, but he will come if something happens. Hope not. We have some other very important persons here today also, and a key person are his supervisors, and his main supervisor is Professor Jane Morell from the Department of Clinical Sciences at SLU, who is his main supervisor. Then we have three co-supervisors. We have Ingrid Hansson from SLU. We have Johanna Lindahl, also from SLU, and now working at the National Veterinary Institute. And then we have Tanapol Nungbua from Maharashakam University.
It's Mahasarakam.
Thank you.
Yes.
And he is not here, or he may be on Zoom.
Yes.
So the order of the procedure is as follows. First, the respondent will give a presentation of the thesis, and then the opponent will initiate a more detailed discussion of the scientific content of the thesis. And after this, we will have a short break. And then after this break, where we will also reorganize us a bit, the examining committee will ask questions and discuss with the respondent. And when you have asked your questions and are content, it will be open so that anyone in the audience can continue and also ask questions and discuss with PAC. And after this, the defense is closed, and the examination committee will meet and discuss. And meanwhile, while they are discussing, we will move to the building over there to have something to eat. And the event today will end with the examining committee presenting their decision. I do not think I will have to repeat anything in Swedish today. I will, and you have no errata list. So then I think we can start this defense, and we will start with your presentation, please.
Thank you, Budu. And good morning, everyone. And for everyone, I have to thank to the opponents and the committees and all the audience to come for my defense today. So the topic today is going to be antimicrobial resistance of the microbiota in the reproductive tract of horses in conjunction with assisted reproduction. Briefly, background about me. My name is Pong Prisha Mala Leung, or you can call me Pak. I graduated from Khon Kaen University, Thailand in 2012 and became a lecturer in Mahasarakham University in Thailand since 2014 and become a PhD student here at Department of Clinical Science at SLU since 2019. So before talking about background on significance of antimicrobial resistance, which is a global issue due to the antibiotic misuse and inadequate infection control, which are also a major contemporary challenge confronting humanity. There are various opinions on antibiotic exposure. Some studies showed that only minimal antibiotic amount can lead to resistance, but some is a controversy about it. And the duration of exposure is also vary between different types of antibiotics. The risk factor for antimicrobial resistant development in the reproductive tracts is the frequent use of antibiotic to treat infections, and there are also non-therapeutic treatment such as insemen extender. The antibiotic insemen extender was used for preserve sperm quality, which was mandated by national and international regulations. The bacteria transfer in healthy saline. Bacteria is reside in mucosa of the distal reproductive tract and can transfer to semen during semen collection or contaminate from the environment during the semen processing. There are limited research on antimicrobial resistance on healthy mares, which not extensively studied when, and they are not remain uncertain about environment impact on environment and anti-cycle on vaginal flora also. And there is no consequence of antibiotic exposure, such as impact of antibiotic in semen extender. There's unclear if changes are temporary or long-lasting. The prevalence of artificial insemination, which are commonly used technique in modern animal reproductions, there's about 50% of foal conceived through AI with chilled or frozen semen during 1990 to 2001. And the more recently study revealed that there are increased use in cool semen for artificial insemination, especially in sport mare, and the growing use for frozen semen in brood mares. However, despite the antibiotic added in semen, bacteria can still grow, which observed in bores, and also in Stallion cement. Bacteria in bore cement unaffected by some antibiotics, but in other antibiotic can prevent bacterial growth. What report in 2014, and in 2015, the first Stallion cement with amikacin containing can found some bacterial growth. The effectiveness of antibiotic is also vary with the bacterial concentration also. And about identification of bacteria, there are two different methods. The first one is conventional culture-based method, which may not detect all bacteria, especially bacteria that not easy to grow on the culture. And there are some limited number of bacteria can identify in some research study. The another identification method is non-culture based, which more recent study employ non-culture based techniques using PCR amplification of 16S gene regions. Over 90% of bacteria genera and 65 to 83% of bacteria species can be identified with 16S sequencing. Then we move on to the managing of bacterial contamination. Antimicrobial agents in cement extender is essential for control bacterial growth. However, there's an alternative emerging such as colloid centrifugation for improved sperm quality and can also reduce the bacterial contamination. This technique is successful in removing 81 to more than 90% of bacteria from Stallion cement. And the more recent study, only 18% of bacteria remain after single layer centrifugation without antibiotics. About single layer centrifugation technique, the technique can separate spermatozoa from bacteria, can improve sperm viability without antibiotic in the cement extender, and it can be practically used in stud farm with access to a centrifuge. There are two types of colloid separation. The first one is high density colloid separation, which can improve the quality. The result is promising for improving cryo survival, but there lost some robust spermatozoa. So there are low density colloid separation, which can use to separate spermatozoa from seminal plasma and bacteria, which retrieve approximately 85% of the original growth in bore spermatozoa without specific selection of sperm robustness. With lower density single layer centrifugation, sperm quality not as well affected by the lack of selection. There was a preliminary AI trial result in pigs that showed similar fertility between control and single layer centrifugation sample prepared with a low density colloid, which this approach can be applied for use in cement, Stallion cement. About the aim of my thesis, the ultimate goal of the topic is an alternative to antibiotic use in cement extender. So the first one is a literature review, which reviewed the existing literature on antimicrobial resistance in the reproductive tracts of males. And the study one, we aim to analyze microbial composition of vaginal flora and their antimicrobial resistance, and also identify the optimal time point for sampling bacterial vagina after insemination. And in study 2, we aim to compare antimicrobial resistance in vaginal bacteria of male before and after insemination. After work with vaginal bacteria, then in study 3, we moving to stallion semen, bacteria in stallion semen. So we aim to investigate bacterial diversity in semen from healthy stallion in the 3 European countries. And in study 4, we evaluate the efficacy of single-layer centrifugation with low-density colloid as an alternative to antibiotic in semen extender for reducing bacterial contamination in stallion semen. The study design for literature review will be focused on antimicrobial resistance in males' reproductive tracts, emphasize on implication of antibiotic use and the alternative to combat antibiotic resistant, which the keyword used is in antibiotic resistant, equine and female reproductive organs. We not include the non-English publications or publication before 1970 and some lower quality studies. In study 1, we divide into 2 parts. The first part is non-inseminated male. Those males come from 2 different places. The first one is 27 males on stud farm that can divided into 3 subgroups, including 2 maiden males, 4 pre-brood males, and 21 brood males. And in other farm, there are 4 other males. Those males were take vaginal sample once, as close as ESTA as possible. In part 2, 10 inseminated male. We taking the swab 4 times. The first time is day 0 before insemination. After we taking the sample, we inseminated those male with semen extender only. And then we collect the sample in the 3 different time points at day 3, 7, and day 14. In study 2, we collect the sample 2 times before and after insemination on 26 males. So in this time, we select day 3 represent for the sample after insemination. We identify bacteria with 5 different agar plates and submitted to MALDI-TOF MS. After that, we select some bacteria isolate for testing the antimicrobial susceptibility using broad microdilution and compare the MIC value before and after insemination. And we select some isolate and submit it to whole genome sequencing to compare whether the resistant isolate before and after insemination is the same strain or not. The bacterial identification, antimicrobial susceptibility testing, and whole genome sequencing is similar to the first study. In study 3, we collect 37 ejaculates from 3 AI centers in Germany, Portugal, and Sweden. The sample were frozen and sent to SLU for DNA extraction. And then we submit the sample to Sci-Lab for 16S RNA sequencing and analysis. And we analyze statistically to compare the difference between Germany, Portugal, and Sweden, which including alpha diversity, beta diversity, and relative SV abundance. In the last study, we collect 18 ejaculate from 6 Stalin semen. Each ejaculate was separated into 3 different groups. The first one is control or extended semen. The semen would add with a semen extender without antibiotic. And the second group was semen, which we prepare similar to the control semen, but we will centrifuge first, and then remove the supernatant and add the new semen extender later on. And the last group, single layer centrifugation with low density colloid. This one is we will add the colloid in the first layer, and then we add the semen, extended semen on top, and then centrifuge with a lower G for 20 minutes, and do the similar as what semen. Then the treated semen and raw semen was analyzed for bacteria count, and the bacteria were identified with multi-top MS. The treated semen also analyzed for sperm quality, which including sperm motility, sperm kinematics, sperm membrane integrity with CARSA, and SCSA with flow cytometry. The analysis is done 4 time point every 24 hours. During that time, we will keep the sample in the refrigerator. Then the result of the first one, literature review, we found that antimicrobial resistance in equine reproductive tract is reported in various countries, which the material mostly form uterus, vaginal, or clitoris swab, which commonly detects antimicrobial resistance. However, is potentially biased in antimicrobial resistant prevalence because of there are more males with problem in reproductive system than healthy males. And we observe the change in antimicrobial resistance over time in some countries, which indicate evolving of antimicrobial resistant pattern. Here is the figure show the global map, which the country report antimicrobial resistant cases within the reproductive system of equine. So you can see a lot of reported in European countries, and there are some reported in the US and some reported in India. In study 1, we identified 41 bacterial species, which E. coli is the dominant species. And we found that there are variation among male subgroups, and there are more diversity in male from non-inseminated group compared to inseminated males. And in antibiotic resistant in E. coli, we found that 24 isolates show resistance more than 1 to 4 tested antibiotics. And there are differences in resistance between inseminated and non-inseminated males. In Streptococcus resistant, we found 2 multi-drug resistant isolates. And there are difference resistance between non-inseminated and inseminated male. For Enterococcus faecalis or isolate were susceptible to antibiotics. And there was a higher number of isolates compared to older time point after insemination. So then we select day 3 represent for time point after insemination for the study 2. And then in study 2, we isolate almost 1,000 isolate from 32 bacterial species. The dominant species is E. coli, Streptococcus, and Acinetobacter. And we found increased resistance to trimethoprim, chloramphenicol, and tetracycline after bacteria exposed to semen extender in E. coli, but not in the other antibiotics. And there's a first case tigersaccharin resistant in E. coli in Sweden reported. And when we look at genotypic cluster, there were increased resistance in some genetic cluster in E. coli. Moving on to study 3. About microbiome diversity, we found about 1,908 ASV, which there were significant variation in bacterial diversity among the 3 countries by Shannon Diversity Index. And there's no significant difference in richness and community evenness among the 3 countries. This figure shows the mean relative abundance in phyla between the 3 countries. You can see the most frequency observed phyla is more than 85% of the total. So this one is similar to the previous study also. Then moving on to the last study. This figure shows the total colony count per treatment group at time 0. So, or the treatment semen is different to each other, which the lowest bacterial colony count is the yellow one in the middle, which come from low density single layer centrifugation sample. And the highest one is extended or control semen, the blue one. This figure shows membrane integrity of the 3 treatmented semen. These are not significantly different, but we can see some lower membrane integrity in the extended semen sample since from 24 hours compared to the other 2 groups. In conclusion about vaginal microbiota in male, we focus on vaginal microbiota compared with the previous literature, which more focus on uterine samples. Our result has high prevalence of E. coli, which attributed to intestinal origin or its rapid growth. The variation in bacterial species might be due to the environmental condition, the housing, agar media, incubation setting, and the identification techniques. And we found that male location influence the vaginal flora, especially on stud farm. And Low-level antimicrobial exposure in semen extender may contribute to antimicrobial resistant development, which we really need more research of effect of husbandry and environmental also. And this knowledge of which bacteria is prevalent could be useful for tailored antibiotic use in the future. About seminal microbiome in stallions across the 3 countries in Europe. The difference in seminal microbiota between the countries is important in tailored antibiotic use for semen extender. The factor affecting microbial load in semen extender might include environmental condition, breeding workload, and abstinence durations. And in the last study, lower density colloid in single layer centrifugation for stallion semen, we conclude the suggestion potential to minimizing antibiotic uses in semen extender. For future consideration of the factor affecting vaginal microbiota, since we found that vaginal microbiota is varied with geographical location, in the future study, we could explore factors like environmental element, seasonal condition, as well as husbandry practice. And about seminal microbiota studies, we could look at abstinence period or bedding types and frequency of changing the bedding, which might impact the bacterial community. Or we can compare microbiota and microbiome together in reproductive tracts for determining the essential antibiotics in semen extender. And about fertility of sperm after low density colloid centrifugation treatment, we could conduct artificial insemination trial to assess the sperm fertility after pass through a low density colloid centrifugation, which we expect not show any issue for male, since there is a lower bacteria after low density colloid centrifugation compared to extended or raw semen. And lastly, I have to thank to all persons shown here, including supervisor, teams, leading by chains, people in Mahasarokam University, in Buden, Vienna, and all the people here in Esalu. And I have to thank to the funding body SVF and my university, Mahasarokam University as well. And thank you all for attention.
Thank you very much for this summary of several years of hard work. Now we are looking forward. Thank you. Now we give the word to Professor Graça Ferreira-Stia, who is going to continue with a more detailed discussion of the scientific content. Please.
Thank you very much, Mrs. Chair. Well, first of all, I would like to greet the PhD committee, Professor Bart Gardella, Professor Maria Kareskowski, Professor Mandy Demestre, who's in US, maybe 4 in the morning there, very early. And I also would like to greet and to congratulate the supervisor, Professor Jane Morel, and the co-advisors, Professor Ingrid Hansen, Johanna Lindell, and the co-advisor from Thailand too. And also to congratulate you for the guidance of this excellent PhD thesis. In fact, being a part of a doctoral committee of the students of Professor Jane Morel gives us a peace of mind because we know the work is of extremely high science quality. So it's really relaxing. It's really a relief, especially also after having all those articles published. It gives us, really a good sense that this is perfect and everything will be okay, right? So it's an honor and a pleasure for me to be part of this jury today in this very prestigious university. It's my first time in Sweden. From what I've seen is a beautiful place, beautiful, nice people, nice colleagues. So I'm really honored to be here. And I also would like to greet all of you who are here today, sharing this special moment with Pak Malalwang. I'm not sure if I pronounced your name correctly. And I would like to greet Pak Malalwang, the reason why we are here today, otherwise I wouldn't be here, and wish him all the best, not only today, but throughout all of your life. So, well, before I start with the questions, that's why I'm here today, I would like to make some comments about this work presented. Well, this PhD thesis focuses on very updated and important topic in both veterinary medicine, but also human medicine, which is antimicrobial resistance of the microbiota. Especially, it focused specifically on microbiota in the reproductive tract of horses in conjunction with assisted reproduction. The work is extremely well presented. The dissertation is very well structured, written, very coherent. I really enjoyed reading it. And Pak, with 5 publications that resulted from this thesis, as well as other 4 publications that I assume you carried on that work while you were here in Thailand, right? The other 4. I assume shows that he has acquired solid theoretical concepts and practical knowledge in this field of research. So this shows he had really dedicated a lot of time to research, that's for sure. I usually say that it's very, very hard to get a P less than 0.05 when you work in science, for common people. That doesn't mean anything, but for all of us, to get that, it's a lot of work, and to publish an article is a lot of work. So we just have to congratulate you and your advisors for all you have accomplished. So your data are very interesting and raise further questions. So I would appreciate you answer my questions if you wish, of course, So if, for instance, you don't understand what I'm saying, please ask again, because that's why I'm here. So in your thesis, you wrote, and very well, that in the 21st century, the World Health Organization has named antibiotic resistance among the top 3 public threats. So this is really an interesting topic. And it says also that according to the United States Department of Health and Human Services, 2019, each year 2.8 million bacterial infections and 35,000 deaths, we're talking about humans, occur in the United States of America as a consequence of antibiotic resistant bacteria. So we should do something about it. And you are on the right track. So you're doing that on veterinary medicine. So you also mentioned that Sweden has a very interesting and a very outstanding approach concerning this issue. Since I'm not from here, I'm really curious about what the policies are in Sweden to avoid antibiotic, bacterial resistance.
As I with a researcher review. So Sweden have annually reported on antimicrobial resistance both in human and in animals. They also encourage people to use antibiotic only for treatment and not allow for like a non-treatment purpose.
Okay.
So, and I'm not sure since when is it the started year, but it's gonna be more than 20 or 30 years now. And it showed that the antimicrobial resistance reported is compared to other countries is a bit lower.
Okay, so for instance, a person here cannot go to the pharmacy and buy an antibiotic.
Yeah.
They cannot unless they have a prescription
Yes.
from the doctor. That's very good.
Yeah.
The same for animals. As veterinarians, only veterinarians are allowed to prescribe and to buy antibiotics.
Yeah.
And they cannot be used for like growth for chickens and things like that. You know, I'm not a chicken person, but in the past, at least, they used antibiotics as growth factors, everything in those intensive productions. So here in Sweden, it's not allowed. The regulations are very strict.
Far as I know, it's not allowed.
That's very nice. It's very good, you know, because not in all countries. I mean, even though there are regulations sometimes that people can, you know, deviate from that. Unfortunately, that's what it is, especially in our, you know, in veterinary medicine. So this concern about antimicrobial resistance was also transferred to veterinary medicine, as you're doing, you know, with your work. And particular to equine reproduction. So you showed us in a very convincing way this is a serious problem. So speaking about bacteria and the mechanisms, how bacteria develop antimicrobial resistance, could you had some explanation in your dissertation a little about it, but could you explain a little more how that could happen? You know, how do bacteria develop that resistance? Are biofilms also involved? Or, you know, could you just tell us some more about it, please?
The development of antimicrobial resistance is according to resistant genes. So the resistance emerging when the bacteria how to say fronting with a harmful environment like antibiotics, a loss of antibiotic in their environments, and then they're trying to survive and mutated or something like that, and they're and the AMR gene emerging, and which those gene can spread to other bacteria close to their strain, or sometimes not close to like both horizontal and vertical transmission.
So they transmit the genes from one bacteria to the other bacteria.
Yes.
So what is the process? How are the genes transferred to the other bacteria?
So for horizontal transmission, there are three different. The first one I can say conjugation. The conjugation is the bacteria can transfer the part of DNA or some plasmid through a specific organ, like we can call pillars, to another bacteria. And there are transformation and transduction. So I don't remember which one is the right name, but they are the way that the bacteria of us can carry the part of resistant gene from the related bacteria to another one. So, and the another way is the bacteria can absorb exogenous DNA through a specific channel. in their cells, and incorporate that into their DNA.
Thank you. Yes, yes. And how can this antimicrobial resistance be avoided? I mean, is there a way we can avoid that,
I
practically speaking?
I think, in my opinion, the way to avoid is try not to produce more.
Okay.
Yes, but there are some cause of metabolics for bacteria that have a more resistant gene. Normally, they cannot survive when they live with other other bacterial flora. So without some dangerous environment, the level of antimicrobial resistance is still constant. But if we not use antibiotic in the right way,
Yeah,
we will speed up
sure.
this to happen.
It's really scary, especially in human medicine, because especially a lot of respiratory diseases with bacteria that are resistant, and there causes a lot of deaths. Anyway, so speaking about veterinary medicine, about your work, among the methods of avoiding the use of antibiotics in semen extenders, you use the single layer centrifugation with low density colloid, but also in the group you work with, you've been using, I don't know you, but Professor Morel's group has used also the high density colloid. So looking at your data and the previous data, what do you think is the best method to treat the semen, to avoid the use of antibiotics in semen extenders? Which method would you choose? When you look at spermatozoa quality, the way of getting rid of the bacteria, eliminating bacteria, which method would you use? The previous method, the high density, or the low density centrifugation?
So
low density colloid.
Yeah, the both high density colloid and lower density colloid had its own benefits, and like high density can remove more. I cannot say more, but can remove lots of bacteria and also can select the better quality spermatozoa.
Okay.
But the amount of sperm left is a little bit lower compared to lower density colloids in the single layer certification. So to adapt to work practically, I can say that we should try to use lower density colloid certification, since they can separate bacteria, spermatozoa from bacteria, and they have no difference on the sperm quality as shown in the study before.
No difference on sperm quality.
Yes.
Okay, thanks. Okay, so you also mentioned that to avoid the use of inappropriate antibiotics in equine semen extender, that it would make sense to identify the bacteria present in semen and then determine the appropriate antibiotic when you cannot eliminate the bacteria just by centrifugation. Would it be feasible in practice in the field? Imagine you're working on a stud farm, you have so many stallions, and you examine their semen, they have bacteria. Would it be feasible or to do this? If a stallion comes in and to the stud farm to breed or to get semen for artificial insemination, how would you do it? Is it practical?
It's practical to apply for single layer certification? Is that?
Yeah.
Okay. So I think it is very practical to use, since all the stud farm they already has a certificate that lots of stud farm they use. If you all remember the watched semen sample, those one They treated cement with centrifugation.
Okay.
So
Yes, sure.
so for single layer certification, we just need to add one layer of colloid, and then we can follow the same thing that the stud farm usually do it.
So, but if you have bacteria that you cannot exclude just by centrifugation, and would it make sense, because you said on page 23 that it would be, should the antibiotic should be, the appropriate antibiotic should be determined. So wouldn't that take a long time to isolate the bacteria? You get the stallion and then you determine the which bacteria are in the semen, which, if they are resistant or susceptible, how long would that take until you get an answer and you could use that stallion?
About the remaining bacteria after the treatment,
Yeah,
I
that's what I'm
think
saying.
I think a lot of technique cannot remove all the bacteria.
Yeah.
But if we look at the result, as for example in fertility trial,
Yeah.
if there are no adverse effect to mares, or
Yeah, sure.
like the fertility rate is okay, I think that could be potential to use this. And in our result, the sample after treatment with single layer centrifugation, they have low number of total bacteria count compared to extended oral semen.
Sure.
That should be a.
So what you mean is that, in those stallions that you collected semen from, most of those bacteria are not harmful to the mares,
yes,
right?
yes.
They're not harmful. Okay,
Yeah.
so it's okay to use it, because from what I read from what you said on page 23, I understood that you're going to determine which antibiotic to use to that specific stallion. That maybe I misunderstood that, what you had written there. So here in Sweden, artificial insemination is used for all breeds. I don't know which breeds of horses you have here that you work with. What were the breeds you work with? Thoroughbreds? No? Quarter horses?
I know that there's like sweet trotter, thoroughbred,
yeah.
also that I used to work with in the I used to work with mare here in Sweden. Yeah.
And artificial insemination is allowed for all breeds, right, in Sweden or not? I'm just curious, this is not your topic, but I'm just curious. Maybe. Okay, maybe.
I will find the answer for you.
I was just curious because it's not the case for all breeds around the world, anyway. So while I was reading your thesis, I was thinking that in the past, people thought, in the medical field, in the veterinary field, we thought the uterus was sterile, the placenta was sterile, semen maybe sterile, everything was sterile in the upper genital tract in females, in males, but apparently it's not true, right? You have bacteria in the semen, so it's not sterile. You have also, of course, bacteria in the vagina. So, and you also, it's very interesting because when you think about the microbiome or microbiota that is essential to regulate many mammalian physiological processes, including reproduction, right? Because not all bacteria are bad, right? Some of them are good. So could you speak a little bit about it, about the presence of bacteria in the genital tract of mares, for instance, if there's a physiological meaning for these flora, if they exist there, why is that? I usually say that nature is very wise. So for some reason, some bacteria exist. Could you speak a little bit about it? Because you did studies on that, right?
Yes, like for the knowledge, as I know, in the reproductive system, there's a lot species of bacteria that can reside on.
Yes.
But as you said, the uterus is the very close system that
Yes.
should be a sterile organs, but as far as my result in the vaginal bacteria, we saw that it's not that sterile. We can found a lot of in the second study found almost 1,000 isolates from there, and those may are healthy. So I think the vaginal flora can can transfer between organ and also environment, like its skin, or come from the feces.
Yes.
In somehow
And what about uterus? The cervix opens up during gastrus, closed.
During as such, yes.
It makes sense if the bacteria can go up. Yes, that's right. So it's not actually in women. There are a lot of studies where they relate different bacteria present in the uterus. For instance, they say that, for instance, Escherichia coli and Enterococcus are present in women with problems with the gynecological problems. But on the other hand, if they have healthy uterus, they have lactobacillus, for instance, that are very useful, very good. They're not bad bacteria. So in women and also in the mares, they have these dysbiosis of microbiota. For instance, if you have a mare with endometritis, most likely, the most common bacteria are Streptococcus epidermicus and Escherichia coli. So you have that also in the vagina. So it makes a lot of sense, that you have bacteria that are useful, others that are not, they are harmful. Anyway, so, and I don't know if you've heard about that in women, human medicine, they even trying to find out if these bacteria present in the uterus could be biomarkers of diseases. Have you heard about that? It's very interesting. If depending on the bacteria, they could predict if the person is going to develop this disease, this uterine disease, or the other one. Mm-hmm. So maybe you're in the right field if you could connect the bacteria that you have in the vagina also with uterus and predict if the mare will develop endometritis, endometriosis, whatever, if she's susceptible. From those mares, did you get biopsies, endometrial biopsies from those mares?
No, we didn't do it.
No, I was just curious, if those mares, did you get different bacteria from older mares versus young mares?
Actually, if we look at the subgroup of mare, they are different in variation, between like brood mare, pre-brood mare, and barren mares. But
yes.
So
you got differences, right?
Got different, but we not calculate statistically, but we look at the pie graph or some figure. So it's not the same.
Not the same, yeah. That makes sense because older mares have more tendency to develop post-breeding endometritis than younger mares. So it makes sense, that's right, So when you talk about the bacteria in semen, you told me that those stallions were healthy, right? So those bacteria are not a concern. So these differences in the microbiota in the semen of stallions from Portugal, from Germany, and from Sweden, Portugal and Sweden were similar. We were very far away, but does it have something to do with the practices on the farm? I mean, here, do they wash the penis before collection or before breeding? Do you have any idea in those how you got similar results? I'm just curious. I have no idea if it's just a coincidence.
Yes, I think
I don't know.
I think that the husbandry can be one of the factors that if the two stud farm in Sweden and Portugal, they follow the same husbandry or similar
Yes,
thing,
yeah.
it might be possible to influence the result that they are close to each other. But how to say, when we look at a Shannon diversity index, all three countries
Yeah.
are different. But in the beta diversity or Baker-Curtis, that we found that Portugal and Sweden is not different. They are similar. So it can say something like that. In some way, they are a similar species,
Yeah,
but in the big picture, it's different.
that's right.
But it's also interesting to look at the husbandry and other factor, like we discussed about abstinent periods also.
Okay. I was wondering if antimicrobial resistance in Portugal is higher than here, probably because unfortunately you didn't look at that, right? Because, the use of antibiotics is, it's terrible, very much widespread, up to maybe some years ago, any farmer could go to a pharmacy and buy whatever he wanted to any antibiotic. In the past years, it's more difficult. Of course, I myself, I can buy whatever I want because I'm a veterinarian. I prescribe,
Yeah, that's
but
similar to my country also.
yeah, so it was very, very common, even the guy at the store would advise, wrongly, of course, selling all kinds of antibiotics, all kinds of hormones, everything. You could buy anything you wanted from those kind of not pharmacies, but kind of supplies for animals where they also had, medicine. But nowadays, with this concern, I hope this, and then again, I don't know here in Sweden, but people have leftover antibiotics instead of going to the pharmacy and leaving there the excess of drugs for them to dispose that. They just throw it in the trash. Just they have no idea, I hope here things are different, maybe also with the farmers. And so maybe I think it would be very important to educate people. I think it's not only to do research, but also educate people and show them in a simple language how dangerous it is, not only for the animals, but for themselves. Maybe then they will understand they're doing wrongly. What do you think?
Yeah, I agree with that thing. So
would you do that? Would you be willing, for instance, when you go back to your country, to do like talks to people, veterinarians, our colleagues? What do you think about that? Extension is very important.
Yeah, I think I will encourage people there first in my university, my student and my colleagues that I work with them. So I will bring the important thing about emerging of antimicrobial resistance and try not to use as we used.
That's right.
Usually did it before.
Yes.
But there's a lot of work to dealing with like farmers, as you say. They can buy everywhere.
Yes.
So there are lots of work to do with like a legislation to control those one.
And the politics in the country. It's important that they do something about it. That's right. Well, this is just anyway, this is a question that I looked at your publications, of course, and I realized you also looked at antimicrobial resistance in the cervix of gilts and sows besides mares. So, Pak, what I would like to know is that could you just talk a bit about the similarities, the differences, what you found in gilt study? How can you compare it to the mare study concerning the reproductive management? Of course, a sow is a sow, a mare is a mare, but the breeding history, the age of the animals, or other factors that could influence. Could you please talk a bit about it?
As far as I remember, first of all, is the difference on vaginal microbiota is different. So that's the first thing. We cannot just solely compare
Sure.
between different species.
Yeah.
Yeah, but we also found some resistance, but
Yes, you did.
as I remember, but not in the major part of isolate.
Okay.
Similar to in.
And also you made a distinction between young, sows and gilts also, between their breeding history, and you got differences. Do you remember that, or you just focus on the mares now?
Yes.
Oh, well, okay. So anyway, but to make a long story short, you also found antimicrobial resistance in the bacteria isolated from the pigs, the sows, the vagina. Okay, yes. So do you think you could also, I'm sure you could also work on that, could also close to the pig farmers, also not to use so many antibiotics, especially in big production. So maybe when you go back to Thailand, you have a role on that too, with the veterinarians, with the producers. I know it's not easy because to change the mentality of people is the hardest thing, right?
I can say that
I don't know how it is in Thailand, but in
even
Portugal,
harder when compared to equine because of we know that there are lots of using of antibiotic as a feed additive or something like that.
Exactly.
Yeah.
That's a big problem. So do you think those antibiotics used as feed additives would also be responsible for antimicrobial resistance?
Yes, I
You
think
think
so.
so?
Because systemic antibiotics also affect the antimicrobial resistance in some local area in the animal organs.
Okay, thanks. Well, this is just a simple question about you work on semen. You did a lot of study on this, and of course you use centrifugation, even to concentrate low concentration sperm. You have to enable you have to remove the seminal plasma and its replacement with semen extender. Why should seminal plasma be removed? Doesn't it have a role in the semen? I mean, just what is the physiological role of seminal plasma in semen? I'm a physiology professor, so I have to ask you this. This is just a physiology, basic physiology. What does the seminal plasma do?
Seminal plasma is how to say, they promote sperm, like they can prevent sperm, and they can a source of like energy or protect sperm in the vaginal or uterus environment, and they are important for sperm to fertilize to egg.
Okay, and also they modulate, seminal plasma also modulates the breeding-induced inflammation in the female reproductive tract before the nutrition of spermatozoa and all that. And it's important because it binds non-viable spermatozoa to PMN and protect the viable spermatozoa in the mare, according to Trodson's work. And also cysteine-rich secretory proteins that exist in seminal plasma also suppress the binding of neutrophils to live spermatozoa. So let the spermatozoa to go to the oviduct, the fertilization site. So you're right, it has a role. So why do you reduce, you eliminate it when during the procedures for artificial insemination?
I think it's because of the way we did single layer centrifugation. Loss of seminal plasma, they cannot pass to the colloid. So that's and and in some case, like to prepare like frozen semen, we need to include semen of plasma as much as possible because it's a effect to the viability of sperm after frozen.
Frozen, yeah.
Yeah.
You have to replace with a semen extender. Okay, so now another question. I'm not a microbiologist, but I realized you use a lot of agar, different agar you use to plate the swabs. You use lactose, purple agar, McConkey, Burt Parker, Deman, and Sharp. You also use the whole genome sequencing for bacterial identification. Could you explain to us why you followed all these procedures? They're correct. I have no doubt about it. But why did you use so many agar? Is that the procedure in microbiology, the normal procedure? When I had microbiology was 40-something years ago. So I don't remember all of these media. Maybe they didn't exist back then. But why did you use all of them?
Yeah.
They have, I'm sure each one is for a different thing.
Yeah. During we are planning for the study, we discuss a lot how many or which type of agar we're gonna use in the experiment. We want as much as possible on vaginal bacteria species
Yes, sure.
that can found. We select some we include some Streptococcus specific, like in the BP Bart Parker agar for that, and MRS agar is a selective media for lactobacillus.
Okay.
And some other I'm not sure remember what the difference of it, but the reason is we should try the most different type of agar for the most variety of bacteria we can culture. And then we use multi-top for identify because it's very convenient to identify. We don't need to compare with the ordinary biochemical test strip that we have a lot of. The multi-top is just what we need just the small amount of colony in the PL culture on blood agar, and we put it in small amount in the metal plates in the multi-top machine, and the machine will read the wavelength of the particle, and they have some data on the machine library, and we can compare those one to the closed bacteria species or genus. And why we use whole genome sequencing?
Yes.
This is the first is after we submitted to microdilution for testing for antimicrobial resistance. We have some question that do we sure that this E. coli, for example, before insemination on this man is the same E. coli isolate after insemination. So then we should do something like whole genome sequencing, and we cluster the isolates that the isolate who share or have similar genetic, we will cluster together, and this is for answer our questions.
Very nice. So from the practical point of view, for a clinician, a veterinarian clinician, what would you do for a diagnosis? Would you go for all of this, or you did this because of research? What about the cost? Is this acceptable for anybody who has a mare to pay for all of this? I'm just wondering, unless some animals, some horses are very, very expensive, cost millions of euros, and, but speaking about a normal mare, normal, I mean, value-wise, Would you go for all of this for just diagnostics?
I think for the diagnostic tool, the
Yeah.
multi-top MS is a good choice for.
Is it?
Yeah.
Good.
For identify the types of bacteria. Yes. I will not encourage using whole genome sequencing for identify because what we need to know is just only what bacteria is the cause of the disease, for example.
Sure.
And then antimicrobial susceptibility test is necessary.
Yeah, that's the traditional one. Yes. So do you have any idea how much it costs per one mare? All of these,
Oh.
I know you didn't pay for it. It was the faculty, but if you have any idea, I don't.
I know that it costs a lot for each sample identification and
because you did a lot of samples.
The very expensive, maybe whole genome sequencing. Yes.
Yes, Professor Morel. Yes, you agree. Okay. Anyway, so looking at your study number 1, you said, and also when you presented, that 41 bacterial species of E. coli collected from the vaginal flora, 58% were resistant to at least one of the 4 tested antibiotics. And AMR also increased in E. coli from mares that were inseminated, particularly they were resistant to trimethoprim and chloramphenicol, and E. coli from non-inseminated mares were mainly resistant to sulfamethoxazole. And you also have streptococcus isolates in non-inseminated mares that showed resistance to erythromycin. And in study 2, you have a similar pattern. You had in 284 out of 520, like 55% of E. coli also showed resistance to at least one antibiotic after exposure to semen extender. This is really frightening because it's a huge, percentage. Could you comment on these findings between before insemination, after insemination? I mean, did they have already antimicrobial resistance before? Are those antibiotics that are much used in semen extender? Or, so please, could you talk a bit about that?
It can be both, like the antimicrobial resistance is already emerging or in that vaginal flora already, or maybe in some cases they are just emerging after exposure to antibiotics in semen extender. But we do not follow or track on the mechanism of this one, but we just can discuss about that. The one reason is there might be some, like when how to say, when at day 0 before insemination, there will be, in some case, there will be lower antimicrobial resistant isolate. But after those vaginal flora contact to antibiotics, the bacteria that sent to antibiotic will disappear. And the one that has antimicrobial resistant genes or can survive and proliferate, duplicate, and increase the number instead. And that might be possible. That's why we found a little bit more antimicrobial resistant isolate after insemination, or compare inseminated mare compared to non-inseminated mares.
When you speak about non-inseminated mares, were any of those mares never inseminated in their life? Or they were not inseminated at that specific season? Or
they are.
Because that would be interesting to look at that in virgin mares, for instance.
There were subgroups like brood mare. We have brood mare in non-inseminated mare. Those mare were not inseminated at least the last breeding season.
Oh, okay.
And we have also some pre-boot mare and maiden mare that never inseminated at all.
Never.
Yes.
So did you compare those to the other ones? Maybe you could look at that.
Yes, we have a diagram showing that, but we are not compare with a we are not compare statistically, but we show the difference of a bacteria species, and it
Could be
it's different.
interesting to look back at your
Yes.
data and see, well, if they were never inseminated,
And it have a higher variety
Yeah.
of bacteria compared to
mhm.
maiden, the brood mare and inseminated mare also.
And what are the antibiotics that you usually use in the semen extender? Because you have different resistances to different antibiotics. Are they all used as a cocktail, or which antibiotics do you use in the semen extender here?
The one I can say about one that we use in study one.
Yeah.
Those antibiotic in semen including penicillin and gentamicin and a fungicide. I don't remember the name, but it's,
Okay.
yes, actinomycin probably.
So then they showed resistance to those same antibiotics that you use in the semen extender,
It's not
or to other antibiotics?
actually, lots of them show the resistance to others rather than that
than
antibiotic
that.
added.
So it means they had contact with the other antibiotics before through semen extender or through generally, systemic administration? Do you have any idea? I know it's difficult to go back.
Yeah, it's
in
difficult
those
to
mares
track that.
to track that.
Yeah, about it.
But it's just interesting.
Yes, yeah. It can be the reason that like the previous treatment can affect or in some reason can be like there are some resistant isolate, but they are not sure like how to say in the whole genome sequencing on study 1 and 2 we found that there